Hello students, welcome to another lecture, Understanding Biology, Chapter 24, Protist. In this chapter, we are going to focus on protist phylogeny and classification. And at the end of this lecture, you should be able to describe how 15 major phyla of protists are related with each other, list the main features of diplomonads, Parabasalids and Euglena, describe and distinguish features of brown algae, diatoms, and water molds, describe the distinguishing features of dinoflagellates and epicomplexians and ciliates, describe the pseudopodia of radiolarians, exterior features of Foraminifera, and how to identify a sarcozoan. Describe major characteristics of red algae and green algae, and finally distinguish between cellular and plasmodial slime modes. So to start this phylogeny and classification of protists, I wanted to start with origin of eukaryotes. Because origin of eukaryotes start with ancestral protists, because protists are the most closely related branch for eukaryotes or ancestors of eukaryotes. So one of the major, most important events of evolution of life is origin of these protists. Eukaryotes actually diverge rapidly in an environment where anaerobic conditions are changing to aerobic conditions. Based on the molecular methods, we now have the ability to put a root to the entire eukaryotic tree. So all known eukaryotes are grouped into 4 kingdoms, as you can see in this phylogenetic tree. So this is the phylogenetic tree where eukaryotes are grouped into 4 main kingdoms, including protists, plantae, fungi, and animalia. So since we are talking about protists, today we are going to look into the ancestors of all eukaryotes. Protists and their phylogeny is very important to understand, because protists are very diverse group of organisms, and it is very important that we know what are the characteristics of this diverse protist group. Kingdom Protista is not monophyletic. They are paraphyletic, and they include 5 subgroups or superfamilies that we are going to talk about. So they group 15 major protist phyla into 5 monophyletic supergroups. There are 200,000 different forms of protist that we are going to talk about. So let's look into the working model of phylogenetic tree of protist. So in this tree you can see these protists are actually classified into superfamilies. So these superfamilies include Excavata here. SAR is a superfamily that have several different groups here, which we will talk about it. And then we have Archaeplastida, Amoebozoans, and Opisthocomta. So these are the 5 superfamilies, and then in those superfamilies we have several subgroups that we will look into through this lecture today. So the first superfamily that we are going to look at is Excavata. This superfamily, Excavata, is composed of 2 monophyletic clades, diplomonads and Parabasalids. So the name Excavata actually referred to a groove down the side of the cell body. So they also have this other group, which we will talk about in a little bit. But what I want you to remember here is that these both diplomonads and Parabasalids, actually monophyletic groups, they share similarities in cytoskeleton features as well as DNA sequences. And we also have another group called Euglena, which we will talk about in a little bit. So let's first look into diplomonads. They are unicellular and they can move. Their locomotion is done by these flagella, as you can see in this picture. One of the most unique characteristics of these diplomonads is that, as the name suggests, they have two nuclei. And these members of this clade can be parasites, human parasites especially. You might have heard about this parasite Giardia. It's actually a diplomonad. And they also, one of another unique characteristic of diplomonads is that they lack mitochondria, but they contain these mitochondria related organisms, or MROs, called mytosomes. So that is one of the characteristics you need to remember, because developing a true mitochondria is very important feature that led to development of eukaryotes. So I told you both Parabasalids and diplomonads are monophyletic groups. So the next one we are going to talk about is Parabasalids. They contain large number of species. They also contain, as you can see in this picture, undulating membranes that assist in their locomotion. And they also contain flagella, which also help them to do their locomotion. One of the unifying characteristics of both Parabasalids and diplomonads is that they lack mitochondria. They also have these mitochondria-like organelles such as mytosomes. This trait is considered to be derived rather than ancestral. Some parabasalids live in the gut of termites and digest cellulose. Some other have symbiotic relationship with cellulose digesting bacteria. So I think parabasalids are quite unique organisms. And the last clade of this excavata that we are talking about, the superfamily excavata, is Euglena. So Euglena is diverged and is considered to be among earliest free-living eukaryotes that possess mitochondria. So one of the important features. Number of Euglena have acquired chloroplast to endosymbiosis. So that is also very important to know. Most of them are autotrophic and others are heterotrophic. So best known phylum of this Euglena is actually euglenoids, which we will talk about in a little bit. One of another characteristic feature of Euglena is they have this flexible pellicle and they do not have sexual reproduction. So when you look at Euglena, one of the, I told you it's one of the best known phylum in Euglena. You might have seen Euglena under the microscope when you look at pond water samples. The members of the euglenoids phylum contain interlocking proteinaceous stripes arranged in a helical pattern, forming a flexible structure called pellicle. So you saw that I told you that they have this flexible pellicle. This pellicle lies within the plasma membrane and provide them the ability to be shifting shapes. So if you ever looked at a euglena under the microscope, you might have seen that they change shape that allow them to pass through different kinds of obstacles. So that is one of the important characteristics of euglena. The reproduction in this phylum Euglena occurs by mitosis, where nuclear envelopes remain intact. You know that in higher eukaryotes, when they go through reproduction, their nuclear envelope breaks, where the centrioles are forming, spindle is forming, and that is normal process. But in these groups, their nuclear envelope actually remain intact, and they do not have sexual reproduction. So Euglena actually contain two flagella attached at the base of the flask-shaped opening called reservoir that is located at the anterior end of the cell. So as you can see in this picture as well. So they also contain another vacuole called contractile vacuole that collect this excess water and empty it into this reservoir. Cells contain numerous small chloroplasts that contain chlorophyll A and B with carotenoids. So concept of single Euglena genus is now being debated. So more the data that we get so far using molecular data, these phylogenies might change later. So concept of a single Euglena genus is now being debated because of that. So second major group within Euglenozoa that we are going to talk today is called Kinetoplastids. They are actually certain parasites, that's why they are very important to discuss here. Kinetoplastids actually contain a unique single mitochondria in the cell. These mitochondria contain an unusual DNA structure called kDNA. This kDNA composed of interlocked minicircles and maxicircles. Maxicircles encode proteins, minicircles encode guide RNA. So one of the popular parasites in this group include trypanosomes. You might have heard about them before. They cause African sleeping sickness, leishmaniasis, and Chagas disease. So you can see they are transmitted by different vectors such as testiflies, sandflies, and skin contact with urine or feces or blood of the infected animal can transmit these trypanosomes into the human bloodstream and cause these 3 different kinds of diseases. When you look at trypanosoma, they are difficult to control because of these organisms rapidly change their protective coat. So why it is very important to know about this control of trypanosoma is because this genetic feature about these trypanosoma is that these testiflies transmitted trypanosomas have evolved an elaborate mechanism for repeatedly shifting the antigens on their protective glycoprotein coat, which now is difficult to cure or treat with normal ways that you actually treat other trypanosoma diseases. And there are many ways that they are actually try to control this disease. They try to release sterilized flies where they cannot reproduce and transmit this trypanosoma, and they actually use traps. And traps, they are actually scented like cows but treated with insecticides to kill the testiflies. And they also now able to sequence the genomes related to these trypanosoma, and then they are hoping actually to find a single drug target to control this disease trypanosoma. Now we are moving into another supergroups that actually called SAR, that include Stramenopila, Alveolata, and Rhizaria. So this supergroup consists in 3 branches. So you need to remember the 3 branches in SAR. And one of the important things about this SAR group is both Stramenopila and Alveolata are monophyletic. They may have arisen by one or more secondary endosymbiosis events. So in this tree you can see that this is the SAR superfamily, and they have 3 branches, Stramenopila, Alveolata, and Rhizaria. So both Stramenopila and Alveolata are considered to be monophyletic and thought to be arisen by one or more secondary endosymbiotic events. So let's first talk about Stramenopila. So in Stramenopila, popular organisms that we are going to talk about under Stramenopila is brown algae, diatoms, and Oomycetes. So one of the characteristic features of Stramenopila is that they have very fine hairs on their flagella, and name suggests Stramenopila means that they have fine hairs on their flagella, and sometimes few species have lost their hairs during the evolution. So let's go one by one. So we have brown algae. Brown algae actually include large seaweeds. One unique feature about life cycle of brown algae is that they have altering generations between multicellular sporophyte that is diploid and multicellular gametophyte which is haploid. Some sporophytic cells go through meiosis and produce spores, and these spores germinate and undergo mitosis to produce large individuals that we see such as kelp. Gametophytes of brown algae are often smaller. They actually, compared to sporophytes of brown algae, their gametophytes are often smaller. Brown algae doesn't contain complex tissues such as xylem for transportation. They are not plants, obviously. The next group is another clade that we are going to talk under Stramenopila is diatoms, which are photosynthetic unicellular organisms with double shells. These shells are actually made out of silica, and these double shells actually look like a small box with a lid, where one perfectly fitting inside the other shell. Diatom chloroplasts contain chlorophyll A and C as well as carotenoids that resemble brown algae and dinoflagellates. They also produce a unique carbohydrate called chlysolaminarin, which is actually very interesting and unique for this group. Some diatoms move by using two large grooves called rapes, and they also have pencil-shaped diatoms that can slide back and forth over the other. As you can see in this picture, diatoms have very pretty structures. This is rape here. They use rape to move, and then pencil-like diatoms use rolling back and forth on each other to move. That is their locomotion, mode of locomotion. And last clade we are going to talk under Stramenopila is Oomycetes, or we call them water molds. And Oomycetes can be either parasites or saprophytes. They live by feeding on, saprophytes actually live by feeding on dead organic matter. They distinguish from other protists by their structure of motile spores or zoospores, which bear two unequal flagella. Zoospores are produced asexually in the sporangium. Oomycetes also have sexual reproduction, which involve producing gametes. Most oomycetes are found in water terrestrials. Oomites are plant pathogens. Some of them are plant pathogens, such as Phytophthora infestans that cause the Irish potato famine. And there are more than 400,000 people died because of the hunger during that disease in Ireland. Now we are moving into superfamily Alveolata. They contain three main subgroups: dinoflagellates, epicomplexians, and ciliates. One trait common for all three subgroups in this superfamily Alveolata is the presence of flattened vesicles called alveoli. These alveoli are stacked in a single layer below their plasma membrane, which may function in the membrane transportation. Let's talk about dinoflagellates. Dinoflagellates are mostly photosynthetic unicells with two flagella. They live in both marine and freshwater environments. And some dinoflagellates are luminous that contribute to the luminous effects seen in the sea or beach at night. Most dinoflagellates have chlorophylls A and C in addition to carotenoids, and similar to brown algae and diatoms, as you remember we talked about them. Some of them are poisonous, and they can create destructive red thrides associated with blooms of dinoflagellates. And their DNA is not complexed with histones, which in higher eukaryotes our DNA is packed with histones, but dinoflagellates, they do not have histones. And these are some of the features of dinoflagellates, as you can see here. And the next subgroup under Alveolata, we are going to talk about epicomplexians. And before that, I forgot to mention that these dinoflagellates actually reproduce mainly asexually. unless there is a starvation occur, then they will reproduce sexually, and during asexual reproduction, mitosis occur in permanently condensed chromosomes that divide within the permanent nuclear envelope. After the chromosome duplicate, their nucleus will divide into two daughter nuclei and then follow their life cycle. Some of them dinoflagellates are actually very interesting. So now let's talk about epicomplexians. The epicomplexians are spore-forming parasites of animals. So the name epicomplexian is due to the unique arrangement of the fibril, microtubules, and vacuoles and other cell organelles at the end of the cell. It is formed as an apical complex. Apical complex is actually a cytoskeleton and a secretory complex that enable them to invade its host cell. The best known epicomplexian is Plasmodium, which cause malaria. Plasmodium actually has a very complex life cycle that involves sexual and asexual phases altering between different hosts. So this disease was most common in Africa, and it was controlled actually focusing on vector control. They use insecticide treated mosquito nests to prevent mosquitoes from biting, and they actually very resistant to treatment or an insecticide, and it actually causes a problem in controlling Plasmodium that causes malaria in Africa. So we also have several other important epicomplexians. So one of them is gregarines. They found in intestines of arthropod, annelids, and insects, mollusks, arthropod, annelids, and mollusks. And they also use their distinct apical complex to attach to the intestinal epithelium of these organisms, and they obtain nutrients through epicomplex attachment from their intestine cells. And we also have Toxoplasma, and these Toxoplasma invade the epithelial cells of human gut using their apical complex. And in immunocompromised individual, this Toxoplasma damage can be in brain, heart, and skeletal tissue. And pregnant women that touch cat litter box can get Toxoplasma through the cat litter box, which is why for the pregnant woman that they do not suggest cleaning the cat's litter box. And last subgroup of Alveolata is called Ciliates, and Ciliates are characterized by their mode of locomotion. They are heterotrophic unicellular protists that contain a large number of cilia. And these cilia can be arranged in longitudinal rows or spirals around the cell. Cilia contain a pellicle. The ciliates contain a pellicle which is thought to be a flexible outer covering that enable them to pass through obstacles. All known ciliates contain two different types of nuclei. DNA in the macronucleus is transcribed for the daily activities, while the DNA in the micronucleus is used only as the germline for sexual reproduction. And they also form vacuoles for ingesting food and regulating water balance, and contractile vacuole regulate their water balance. And one well-known example of a ciliate is called Paramecium. So here is an example picture of Paramecium. And Paramecium can undergo sexual reproduction through conjugation, which allow them to exchange their micronucleus. Now we are moving into another superfamily called Rhizaria. And this Rhizaria superfamily have armoporous shapes with protruding pseudopods. They are roughly characterized as amoeboid. Members of the phylum Actinopodia called radiolarians secrete glassy exoskeletons that's made out of silica. These exoskeletons can either be bilateral or radial symmetry, and the shells of these different species have beautiful shapes with pseudopods extruding outward. So they actually have three distinct groups in superfamily Rhizaria. They are Radiolaria, Foraminifera, and Cercozoa. So let's look into Foraminifera as the first subgroup under Rhizaria. So phylum Foraminifera actually contain heterotrophic marine protists. They can range in diameter from 20 micrometer to several centimeters, and they contain pore-studded shells composed of organic material reinforced with grains of calcium carbonates, especially sand. Depending on the material they use for the shells, Foraminifera can have shells of very different appearance. Most live in sand or are attached to other organisms, and their life cycle of Foraminifera can be extremely complex, which alter between haploid and diploid generation. Now we are moving into another superfamily, which is Archaeplastida. They contain four subgroups. They are Rhodophyta, Chlorophyta, Charophyta, and land plants. We will look into Rhodophyta, Charophyta, Chlorophyta in this lecture. So both red and green algae make up the fourth protist supergroup, and both red and green algae are thought to be descended from a single endosymbiosis event over 1 billion years ago. So this supergroup is important as it indicates the direct evidence that support that the protists actually closely related to plants, especially for green algae. So the plants are actually evolved from green algae. This supergroup is actually important to show that connection. So let's first look into red algae. Red algae are photosynthetic, multicellular marine algae. There are 6,000 described species in this phylum, indicate common ancestry with green algae. Their red color is actually due to the presence of accessory photosynthetic pigment called picoerythrin. Picoerythrin together with accessory pigment picocyanin and allopicocyanin are arranged within the structure called picobilisomes. They allow algae to absorb blue and green light at greater depths. Their size vary in microscopic to single cells organism to multicellular seaweeds. So these are some of the examples of seaweeds, kelp, seaweed that you might have seen when you went to visit the sea, and they're actually very unique seaweeds. Now we are moving into Chlorophyta. So green algae have two distinct lineages, Chlorophytes and Streptophytes. Streptophytes actually contain Charophytes that gave rise to the land plants, and many Chlorophytes are unicellular, and these earliest green algae probably resemble Chlamydomonas. And Chlamydomonas, are microscopic, green color and rounded, and they have two flagella at the anterior end. Most individuals of Chlamydomonas are haploid. They reproduce asexually, and their life cycle is not haplodiplontic. So we will look at what is haplodiplontic in a little bit. They also can actually be very unique depending on how they reproduce. They can also reproduce sexually as well. Then we have multicellular chlorophytes. The Chlamydomonas is a single cellular chlorophyte. So colonial multicellular chlorophytes have some cell specialization. They show cellular specialization, which is an aspect of multicellularity. This developed a specialized motile colonial organisms. They are unlike single cell or Chlamydomonas, volvox actually considered to have many cells in them. They have hollow sphere that made out of single layer of 500 to 60,000 individual cells, each cell having two flagella. Only a small number of cells are reproductive, and some reproductive cells divide asexually, bulge inward, and give rise to a new colony. So that is, here is a picture of volvox, and their reproductive cells produce new colonies, which you can see here. And let's look into haplodiplontic life cycle in multicellular chlorophytes. So multicellular chlorophytes have haplodiplontic life cycle. One of the example of multicellular chlorophytes are Ulva that have haplodiplontic life cycle. So haplodiplontic life cycle is actually very unique. It's different than diplontic life cycle. So in haplodiplontic life cycle, multicellular haploid and multicellular diploid stages can be seen. Actually, gametes develop in the multicellular haploid gametophyte. They fertilize these gametes and give rise to multicellular diploid sporophyte, in which these haploid spores are produced, and these spores can give rise to the organism later. So this actually differs from diplontic life cycle, which only the gametes are in haploid state. So I told you one example of multicellular chlorophyte that has haplodiplontic life cycle is Ulva. They are multicellular chlorophytes. They have identical gametophyte and sporophyte, and they consist of flattened sheets of 2 cell thickness. So further, when you look at the life cycle of Ulva, they are multicellular diploid stage, which is called sporophyte, produce haploid spores by meiosis. And diploid spores, mother cells, actually undergo meiosis in sporangia. They produce spore haploid spores, and then they also have multicellular haploid stage gametophyte. These spores divide by mitosis, produce gametes by mitosis. These gametes fuse to form diploid zygote. So this is how the life cycle of Ulva is actually going through haplodiplontic life cycle. And I told you one of the most close related to the land plants is chlorophytes and charophytes actually. So charophytes are the closest relative to plants. It's a clade of streptophytes. They are also green algae. They are distinguished from chlorophytes due to their close phylogenetic relationship with plants. So here is two members of charophytes. We have chara and coleochaete here in this picture. So charophytes actually very interesting organisms. They are distinguished from chlorophytes, I told you, because they are close relationship with the land plants. And the evolution of diplontic embryo and haplodiplontic life cycle occurred after they moved to the land. Actually, because charophytes actually have haplontic life cycle. So I told you there were two candidates in Charophyta clades, Charales and coleochaetes, and both Charophytes clades form green mats around the edge of the freshwater ponds and marshes, and one species must have successfully inched its way into the land through the adaptation to drying out. So this is one of the landmarks for having the flowering plants and land plants that we have today. One of the important events that happen. Now we are moving into another superfamily, which is called Amoebozoa. So Amoebozoa are actually Amoebozoa and Opisthokonta, which contain fungi and animals, are very close related, as you can see here in the phylogenetic tree. We are going to first talk about Amoebozoans, and then we will talk about Opisthokonta in a little bit. So Amoebozoa is the last supergroup of Protistean tree. They actually move using their pseudopodia, and these pseudopodia can form at any point on the cell body of their Amoebozoas, and they are also called slime molds. Like water mold, they once was thought to be fungus, but they are not actually fungus. They are protists. There are two distinct classes of slime molds. So we will talk about them in a little bit. Another important characteristic of pseudopods is that these pseudopods help them to engulf their food particles, and they actually put the pseudopod forward and then flows into it. If you have ever seen amoeba under microscope, their movement is very interesting. So most amoeba are free living. They found in soil as well as in fresh water, and some of them can be parasitic. If you have ever heard about brain eating amoeba, it's a parasitic amoeba that can enter through your nasal passages, go into your brain and eat your brain, literally. And then we have acanthamoeba that enter to the body through wound and cross the blood-brain barrier and go into the brain and damage your brain. So I mentioned that there are two distinct classes of slime molds. We have plasmodial slime molds and cellular slime molds, which previously considered to be fungi, and we are going to learn about these slime molds. So plasmodial slime mold, the difference between plasmodial slime mold and cellular slime mold is that plasmodial slime molds are huge, single-celled, multinucleate, oozing masses, while cellular slime molds are actually cells combined and differentiate, creating an early model of multicellularity. So let's first look into plasmodial slime molds. They are large single-celled multinucleate oozing masses, as I told you before, while cellular slime molds are single cells. So plasmodial slime molds can stream alone as a plasmodium. There is a non-walled multinucleate mass of cytoplasm. This form is called a feeding phase. As they move, they can engulf and digest bacteria, yeast, and other organic matter. Multinucleate plasmodium cell can undergo mitosis synchronously, with the nuclear envelope breaking down at late anaphase or telophase. When either food or moisture is scarce, they migrate to new area and produce mature sporangium to produce spores, which is the spores can be transmitted and produce another plasmodial slime mold elsewhere. And then we have cellular slime molds. They have relatively simple developmental systems. Individual organisms behave as separate amoebas moving through the soil and ingesting bacteria. Again, when the food becomes less available, these individuals aggregate to form a moving slug. This slug goes through a morphogenesis to produce a stalk and a spore cells. Then the spores form new amoeba if they land in a moist habitat, this way that they can find new niches for populate. And lastly, we are going to look at a group of protists that is likely to be our animal ancestors. Like Charophytes, Chara is our plant ancestor, land plant ancestor. The protist group that belong to superfamily Opisthokonta is actually considered to be our animal ancestor. This group is called Choanoflagellates. Choanoflagellates are actually more like sponges. They have single flagellum surrounded by a funnel-shaped contractile collar composed of closely paced filaments. So, choanoflagellates are considered to be the animal ancestors that we see today. So, they are actually very important. They also feed on bacteria strained out of the water through their collar. And then they also have colonial form that resemble freshwater sponges again. And they have also cell surface receptors. You might have heard about this cell surface receptor, which is tyrosine kinase. And these tyrosine kinase actually show strong homology found in sponges. So there are many evidence that show that choanoflagellates are very closely related to sponges. So that is the end of the lecture. So in summary, phylogenetic tree of protists contain the ancestors of plant, fungi, and animals. Diplomonads lack classic mitochondria but have mitochondrial related organelles. They are unicellular and also have 2 nuclei. Parabasalids also have MROs. They use flagella and undulating membranes for locomotion. We have Euglena. They are among the earliest protists to contain mitochondria. Most members of Stramenopilla have fine hairs on their flagella. And brown algae are large seaweeds that undergo alternation of generations. We have diatoms that are unicellular with cilia in their cell walls. Oomycetes are unique in production of zoospores. Alveolata have flattened stacked vesicles below their plasma membrane. Dinoflagellates have paired flagella and swim with the spinning motion. Epicomplexians are spore-forming parasites and ciliates are unicellular. You have red algae that vary greatly in size, have accessory pigments that give them red color. Green algae have chloroplasts that is very similar to plants. And plasmodial slime molds consist of single large multinucleate cell, while cellular slime molds are multicellular. Hope you enjoy this lecture. Until we meet with another lecture, have a good day. Thank you.